Thank you very much. Happy to be with you this afternoon. Today, for this new episode of the RedTalks by Kalim, happy to co-chair this session with my friend and colleague Sylvain Carras.
Hi.
From the University of Grenoble. I'm Loïc Ysebaert from the University of Toulouse, and today we will discuss the metabolic reprogramming in chronic lymphocytic leukemia, and we will discuss both the T and the B cell perspective, because today we have the chance of having Professor Eric Eldering. He's a professor of molecular immunology at the University of Amsterdam in the Netherlands. Eric has been working on apoptosis for long years. He has a long track records on apoptosis in CLL, but also in normal immune cells. And now his work has turned a little bit to the study of immunometabolic perspective in this disease. And so I'm happy to leave the floor to you, Eric, and we will have a time for question after your talk. So thank you again for accepting our invitation and spending an hour and a half with us today.
Thank you, Loïc, and the organizers for the opportunity to present the work. I believe the title is already on the screen.
Mm-hmm.
So indeed, I would like to tell you about our work on metabolic reprogramming in chronic lymphocytic leukemia, both from the CLL perspective, from the CLL cells, and also the T cells involved. So there will be quite a few different aspects, I think. I would like to stress that the last part of the work on the T cells and associated stuff is not published yet. So I was just thinking we have to figure out a way to organize that with public exposure on YouTube, but we'll talk later about this. Let me see if the slides work. Yes. So if we start, some general notes on metabolism. Here are the cancer hallmarks as defined and widely known by Hanahan and Weinberg, and an important aspect of the changes that cancer cells undergo are changes in metabolism. This has been known for a very, very long time.
Sorry, Dr. Eldering.
Yeah.
We can't see your screen. Can you share it, please?
You cannot. Oh, I thought we were already sharing. Sorry.
No problem.
No problem.
Like this?
Yes,
That's
I
better?
can see it. I will check for the spectators.
It says on my screen that I'm sharing the screen.
Yes, it's good.
It's all good. Sorry for the perturbation.
Okay. We'll continue. I was talking about this slide where the changes in metabolism have been studied for a very long time, and especially with a phenomenon that's called the Warburg effect or the Warburg dogma in cancer metabolism. And the Warburg dogma states what is written here, that all cancers primarily execute glycolysis, which means the use of glucose and convert it to lactate, even when oxygen is sufficient for the cancer cells. So this was called aerobic glycolysis. So this dogma has been challenged since about, I would say, 15 years perhaps, that it does not hold for all cancers, and that actually a new phenomenon called glutamine addiction has also taken a center stage in cancer metabolism. And we will also show data that this may also hold for CLL. But that will come later. I'm not aware to what extent the audience is familiar with CLL, but here are some basics. So chronic lymphocytic leukemia has been actually quite successfully treated with targeted drugs, but it's still considered an incurable malignancy, and patients suffer, apart from the leukemia, of course, from immune dysfunctions and infections. So that was for us a reason to also look into the T cells of CLL patients. Important to know is that CLL, though called a leukemia, the active disease actually takes place in the lymph nodes of the patients. And there is actually where the disease is, there's where the cell division takes place due to B cell receptor triggering and various signals from the tumor microenvironment, both from surrounding cells, cytokines and chemokines. This induces a pro-survival state and expression of pro-survival proteins, which we have studied in the past for quite some time, and this will be also sometimes in the slides that I will show you. Important to note also that this cell activation in the lymph node also causes a metabolic reprogramming from inert state in the blood to active state in the lymph nodes, and this poses challenges if we want to study CLL, because we take the cells from the blood, but there they are inert. They are not cycling. They're highly sensitive to drugs. For example, venetoclax, you can easily kill cells also in the laboratory by venetoclax, but the cells in the lymph node are resistant. We will show examples of that. Just in passing, so the bone marrow is also a repository of CLL cells in the patients, but the bone marrow, although has been studied for quite a while and is used in diagnostics, is actually not a site of active disease. So it's primarily lymph nodes or spleen where that takes place. Perhaps important notion to realize. Now, still part of the introduction, some key points on metabolism in B cells, but also T cells. They are listed on the bottom of the screen. Can you see let me see. Do I have a pointer? No, I don't. Okay, can you see my pointer?
Yes.
Okay, so here are some notes that in non-malignant cells, from naive B cells, germinal center B cells, activated B cells, plasma cells, all these different states use distinct fuels for their energy metabolism. So glucose, as I've noticed, is used for glycolysis. Oxidative phosphorylation takes place in the mitochondria in a process called the TCA cycle, tricarboxylic acid cycle. And this is important to realize that this TCA cycle can be fueled by glucose, fatty acids, and amino acids such as glutamine. We will touch upon these later in the talk as well. And also important to realize, I think, that CLL cells in general, especially the lymph node state, so where they are active, mostly seem to resemble the activated B cell state. So the outline of the talk, as I prepared it today, is first a part on the CLL cells, the analysis of metabolic reprogramming that we did in the CLL lymph node tumor microenvironment, leading to a little bit deeper into glutamine addiction in the proliferating CLL. Then we switch to the CLL T cell dysfunction, as I've mentioned, that exists, the metabolic aspects of that, and also the difficulty in studying T cells in CLL leukemia, because the large majority of cells in patient blood are in fact CLL cells, so it's difficult to study the T cells. But our group, not so much me, but our group has become pretty good in that. And I would like to end with a practical application based on our work on glutamine importance for CLL. We are developing labeled glutamine as a PET tracer, and I hope to end with that if time allows. So here's a sort of scheme that we developed to illustrate how we want to study the CLL cells as they occur in the lymph node. So we used for that patients treated with ibrutinib, which causes the cells to move from the lymph node to the peripheral blood, as is illustrated over here. So, and these samples we obtained from patients at baseline and after 3 months ibrutinib were studied in vitro in the laboratory, either unstimulated, CD40 stimulation, or B-cell receptor triggering. And these are in vitro manipulations of the cells in order to render them into a lymph node-like state. That was the idea. We've been doing that in the lab for quite a while. And by using and comparing these cells with ibrutinib treated patients, it's now going. We were hypothesizing that the ibrutinib treatment will suppress the metabolic activity of the CLL cells, and this will be mirrored by the CD40 B-cell receptor stimulation. And we used in first instance metabolomics, where you measure the quantity of about 100 metabolites in the cells to find support for this premise. Later on, we included RNA-seq and 13C tracing, which I will get into. And this was all analyzed by various sets of bioinformatics. So to come to the first results based on the metabolomics, here we have a quick shortcut to the confirmation of our hypothesis that the ibrutinib treatment versus, and on the top, the CD40, or the bottom, the B-cell receptor stimulation that we did in vitro, indeed has the opposite effect on the metabolic pathways that seem to be active or inactive based on the metabolites, because the direction of the various pathways involved goes in opposite directions. And if we summarize then what is true for both the CD40 and B-cell receptor stimulation, we find that these pathways in this rectangle over here were common and seem to have large effects in the CLL cells, and also were downgraded if cells were treated with ibrutinib in the patient. And from these, already 2 metabolites and fuels of the various energy pathways seem to be very important. So glucose, not surprisingly, but also glutamine and the glutamine metabolism seem to play an important part in the lymph node setting of the CLL cells. So we, in the next set of experiments, we go deeper into the role of glucose and glutamine in the CLL cells. First, we did the RNA-seq, so bulk RNA sequencing, and this is a quite busy slide, but we already filtered out various pathways. For example, on the right here, you see glycolysis and the genes involved, and the samples that we picture over here are average Z-values of the gene expression in the various samples that we studied. And we see here the B-cell receptor stimulation, the CD40 stimulation, which we did in our lab, and ibrutinib treatment for 1 or 6 months, as was published by the group of Dan Landau in 2017. And here on the far left, you see the actual lymph nodes of patients, and that was derived from an earlier study based on microarray effect from lymph node data. So we managed to recalculate these data and compare them. And the purpose of this representation is actually to see whether the lymph node, for example, over here, these genes are indeed all upregulated, or most of them, via B cell receptor or CD40 stimulation and downregulated in the peripheral blood as analyzed by the group of Landau. And this again confirms our hypothesis that in order to study the lymph node, which is on this, we can use in vitro stimulation by B cell receptor triggering or CD40 triggering in the laboratory. So this validates our premise. So we continued using that B cell receptor or CD40 stimulation as a proxy for metabolic reprogramming in the patient's lymph node, because it is in practice very difficult to obtain lymph node cells, viable lymph node cells from patients. So that's why we want to do laboratory studies for that. So also the same is true for genes involved in oxidative phosphorylation. You see the same scheme. So ibrutinib turns genes off and the in vitro stimulation turns them on. And like I said earlier, we would like to focus on glutamine and glycolysis. So we've highlighted here some genes involved in glutamine metabolism and glycolysis over here. And this started our interest in genes with complex names and numbers. We call them SLUC1A5, and these are in fact glutamine transporters. So the cell membrane transporters that mediate the import of glutamine from the blood or the medium into the CLL cells are highly regulated. And we thought it was very interesting, and we wanted to study this further. And this piqued our interest of glutamine as an energy source in the CLL lymph node. And to add to that, what you see here is actually the glycolysis genes. And this one over here, SLUC2A1, is actually GLUT1, which is the importer of glucose. And what you can see that this actually turned down in the lymph node and after B cell receptor stimulation, and it goes up in the peripheral blood, showing that the cells in peripheral blood mainly depend on glucose, whereas in the lymph node they seem to depend more on glutamine. And we continued studying that and finding support for that premise, and also trying to figure out whether we can use this as a therapeutic or diagnostic tool. In order to confirm this, we did so-called 13C tracing, where you used radiocarbon labeled glucose or glutamine, and the experimental scheme is depicted over here. We have unstimulated cells, CD40 and B cell receptor, again as a proxy for the lymph node setting of the CLL patients. And either you add for 2 hours labeled glucose or glutamine, and after 2 hours the cellular lysates are analyzed by liquid chromatography and mass spec. And for example, over here is one metabolite from the glucose pathway, and if you stimulate the cells and feed them glucose, the incorporation of carbons in this metabolite, UDP hexose, is very strong, and it's much less strong if you feed the cells glutamine, which is logical because this is glycolysis intermediate. And a bigger scheme is in the next slide. So the full picture here was the metabolite that we just showed. So it's high up in the glycolysis pathway. Here is the TCA cycle with its metabolites, citrate, alpha-ketoglutarate, succinate, fumarate, and so forth. May be familiar to some of you, but the detailed knowledge of this metabolism is not required, but it's what is important to just look at the orange or green bars, because orange means glucose plays a role and green means glutamine plays a role. And you can see that the TCA cycle, so the mitochondrial energy household of the cell, is mainly fueled by glutamine, because you have to realize that in the circumstances where we measure glutamine radiocarbon, there is also glucose present. but it's mostly glutamine that gets incorporated, as opposed to up here in the glycolysis, where of course the carbons come from glucose. So more details on that is also, we found that glucose is fueling what is called the pentose phosphate pathway, which is generating building blocks for DNA. And glutamine is more involved in glutathione metabolism, but that's a detail that's not very important now. In summary, what we've found is that glucose is fueling, of course, the glycolysis pathway, and the pentose phosphate pathway is an important aspect for dividing cells. And of course, the Warburg effect is existing also in the CLL cells, but in the mitochondrial metabolism, also very active in the lymph node setting, as we studied it. The mitochondrial pathways are fueled by glutamine. So, as a side conclusion, we can say that the CLL cells do not conform fully to the Warburg dogma, as they do not fully depend on glucose. Okay, I'm switching gears a little bit here, and we have set up a spheroid 3D CLL setting, of which you can see some movies over here, unstimulated cells on the left. And when we culture these cells in what they are called ultra-low attachment plates, or ULA plates, and we give them a cocktail of CpG, which is a TLR9 ligand, and cytokine cocktail, IL-2, 15, and 21, you can see that the cells start growing and they form follicular sort of substructures. And this is actually a very useful model also as a lymph node proxy, which we've been using in the recent years. And it is, as far as we know, the only system where the CLL cells actually proliferate a lot, which is in the laboratory difficult to achieve. If we now look on the right, and I start the movie there, this is the growing or proliferating CLL cells, but now in the presence of a compound called V9302. And that is actually what it is a specific inhibitor of this importer of glutamine. So now the import of glutamine is blocked, and this has immediate effect on the CLL cells, because the growth is really prevented in this system. And we've been using this compound, which has been developed actually for therapeutic purposes and has been tested in mice. We've been using this as a tool a lot in our studies. What we also did, besides the effect of glutamine incorporation into the CLL cells, we've also studied its role in protein synthesis and resistance to venetoclax. I've said in the beginning that CLL cells are highly sensitive to venetoclax, which has been used in the clinic. And this is depicted here in the middle with the black lines. So nanomolar concentrations already after 24 hours strongly reduce viability of the CLL cells. And the system we've been using a lot in the lab, I mentioned, is CD40 stimulation. If we do that for 24 hours, provided the CD40 ligand is provided by murine 3T3 cells expressing a human CD40 ligand, you see a tremendous increase in resistance to venetoclax. This is a known phenomenon, at least it was published by us and various other groups. And on the right in blue, you see that also B-cell receptor triggering, though less strong, provides protection against venetoclax treatment. And the interesting thing here is the first time that we use this V93 inhibitor is that it reduces this resistance. And this was for us an indication that the glutamine usage of CLL cells is in fact also important to synthesize pro-survival proteins. And we studied this for a bit. Okay, I will come to that later. We wanted to now compare the V93 inhibitor with just glutamine deprivation. So, and then we noticed, if we do again metabolomics and amino acids, that the inhibitor causes a pronounced decrease in most amino acids, and the glutamine deprivation, as expected, mostly impacts the glutamine-derived pathways, which is depicted over here. So the V93 reduces many other amino acids, whereas the glutamine depletion from the medium has effects on just a few, glutamine, glutamate, aspartate, as expected. And why is this important? Because we wanted to study what does this V93 actually doing to the cells in order, for example, to reduce the resistance to venetoclax, as we've just seen. And in order to study this a bit more, here's a scheme of what glutamine is used for in the CLL cells. So glutamine is first enters the cells via this transporter, is then converted to glutamate, and can be used to synthesize other amino acids. I've shown you already that it is used to fuel the TCA cycle, and glutathione is also produced from glutamine, but it's also known to contribute to synthesis of nucleotides and protein synthesis in general, of course. And in order to figure out which of these is the most important, we started studying this. And just to make the talk a little bit shorter, the glutamine import into the TCA cycle via alpha ketoglutarate was not limiting, and neither was the glutathione, neither was the nucleotides. So we could sort of strike away all these options, and this left us with protein synthesis and other amino acids, and I will show you some data on that in the next few slides. And here's again a scheme to illustrate what we slowly started understanding, what this blocking of glutamine import does to protein synthesis. And for that, here's a scheme of various transporters involved in amino acid import. Here's the glutamine importer that I mentioned. The gene is called SLC1A5, and the protein is called ASCT2. So it's quite a complex world. All these importers have gene names and completely different protein names. What is important from this slide is that when glutamine is imported by this transporter, the SLC1A5, once it is inside the cell, it can be exported again. And this is a known phenomenon because glutamine is used as an exchange factor to import leucine. And leucine has been described to be very important for mTOR and protein synthesis because the leucine levels in cells, they determine the activity of mTOR. mTOR is very important protein complex for protein synthesis and in cells. So that might be an explanation why blocking this receptor in fact has effects on mTOR. So now we wanted to study, if we use the V9302, what are the effects on mTOR activity and protein synthesis in the cell. So these are the next couple of experiments. Again, we use the system of CD40 stimulation in this case. And a readout for mTOR activation that is used in many groups and many laboratories is phospho-S6 readout. So if we have the peripheral blood CLL cells stimulated by control cells there's not a lot of phospho-S6 because mTOR is not very active in these peripheral blood CLL cells. If we stimulate them via CD40, activity goes up. If we deplete glutamine or we use the inhibitor, the activity goes down again. And as a control, we use rapamycin, which is an inhibitor of mTOR, and that is expected to go down again. So indeed, V93 and glutamine deprivation have strong effects on mTOR activity, probably via this leucine exchange, which I just mentioned to you. If we now want to study the protein synthesis as under the same circumstances, we can use an assay for fluorescent labeling again the same conditions, and we see again that protein translation in general is also prevented if we add the inhibitor, and slightly less so if we just deplete glutamine, which is to be expected based on the previous results of the metabolites. And again, rapamycin also prevents protein translation. And as an illustration of that, we use the MCL1, which is again strongly induced at the protein translational level if we use CD40 stimulation. This is known from other groups and our own data. And we can indeed lower the levels of MCL1 by using V93 and again less so glutamine deprivation, which can explain the previous results that I showed you for the venetoclax resistance, which is in fact mediated partly by this pro-survival protein. This is known. I hope you are still with me with lots of these data. Thanks for sticking with me at this late hour. Here are some data again on the role of glutamine for the CLL proliferation, switching back a little bit. So we now compare the 3D versus the 2D settings, the spheroid system on the left. And here is the 2D system that we've used in the past a little bit, where we stimulate the cells with CD40 stimulation plus IL-21. This makes the cells proliferate to a lesser extent than in the 3D system, but this is the system we used in the past. And if we now add the inhibitor, as I've shown you, in both systems, there is a complete block of proliferation. And this tells us that the effect of the V93 in the 3D setting is direct on the CLL cells and is not via the T cells that are also in there. And this is important as a little bridge to the T cell studies I will be showing you next, that these do not depend so much on glutamine as the CLL cells do. So this gives us perhaps in the future a therapeutic window to target the glutamine dependency of CLL cells, while we then do not affect the T cell proliferation, of which we have an example here on the right. If we stimulate CD3, CD28, and in the presence of this glutamine importer, this does not prevent the proliferation of the T cells in this system. Okay, this brings me now slowly to part 2 of what I want to tell you. And I will speed up a bit because I see the time is progressing. What I want to show you, and I'll speed up a little bit here, is that the metabolic dysfunction in CLL T cells are based on mitochondrial defects. And I will maybe skip this part for the sake of time, because it's more a technical matter. So we'll get to that later. And here below are the names of the people who actually did the work, which is technically sometimes quite complex. So again, the CLL characteristics, and now with the focus on T cell dysfunction in CLL. The T cells, and it's known by studied by various other groups before we started working on this, is that the T cells have impaired proliferation, defective signaling, decreased functionality, and a skewing towards effector phenotype and not so much memory phenotype. And memory cells are T memory cells are quite important for immune function. Some first indications that the mitochondria of CLL T cells, as we call them, are altered is just looking at unstimulated day zero baseline, and we can see that the mitochondrial polarization of the CLL T cell mitochondria is reduced. And this is true for the various T cell subsets. This is important to mention because as you can see with these population is that effector memory populations in CLL cells are skewed more towards effector. But the differences are true for all these subpopulations. So it's not based on skewing of the T cells in CLLs. That's an important notion. If we then continue, and now we want to do metabolic profiling of CLLs by seahorse. This is a fairly well-known machine these days in the labs, and it's basically uses the measurements of lactate production and oxygen consumption as proxy for glycolysis and oxidative phosphorylation. I think this machine has become very popular in labs all over the world. It measures acidification here on the top and oxygen consumption here on the bottom. And this reflects the mitochondrial activity and glycolytic activity. During the measurements, various inhibitors are used, and this gives all kinds of information on mitochondrial activity and respiration or glycolysis, as these graphs on the top illustrate. I won't go into the details of how this works. It's important to realize is that basal glycolysis over here is not significantly different, but is widely varying between in CLL samples. Each circle is a CLL sample. And what you can see is that the basal respiration, so the mitochondrial activity in CLL patients, is generally decreased. And actually, we have two types of dysfunction in the CLL T cells, and that is cells that seem to be stuck in a quiescent state and are not activated at all. There are CLL patients that do show some increase in glycolytic activity, and compared to healthy donors, they make a switch to a combination of glycolysis and oxygen consumption in a 2-day stimulation assay. So we have two types of dysfunction in CLL, cells that are actually hardly activated in these conditions, and cells that become glycolytic but don't make the transition to the increase in oxygen consumption. And this is a more detailed depiction of that, and it's true for all kinds of aspects that you can measure with the seahorse machine. We will go a little bit quicker. And here on the right, you see this. So there's cells that, if you look at the CD25, which is an activation marker of the T cells, there are cells that are permanently low and cells that actually seem to be normal in that respect. But the mitochondrial activity, as you can see on the left over here, is decreased in both groups. So there's something wrong with the mitochondria in CLL T cells. And it's important to know that what is here in the box, it seems to be a stable phenotype per patient. It doesn't really matter if we take blood 3 months later and it's still the same. So it seems to be fixed per patient. And we don't know what is actually the cause of this, but this is our observation. In view of the time, I will skip this. This is just to say that the summary of 13C tracing, actually highly complex studies by the group, but I will just summarize it by saying that the fueling, so the entry of glucose glutamine or fatty acid into the mitochondria is not the cause of this defect. So we could strike that out. One thing that was then the next thing that was studied by the people in the group is actually the mitochondria in the cells, and just looking at the shape of the mitochondria. And this is done by computer-assisted image analysis. And there's various ways that the mitochondria can then be projected from a 3D state on a 2D image, like you can see over here. But even just by eye, you can already see that healthy donor mitochondria, here fluorescently labeled, are quite different from CLL T cell. So this is not a CLL cell, it's a T cell from a CLL patient. And you can already see that the mitochondria have a different shape. And there's various ways the computer can analyze that, but all the aspects of mitochondrial shape, the elongation, complexity, and the branch length that they have, they're all decreased in CLL. So there's a morphological difference, which may be connected with the OXPHOS defects that we see. Although, so I say here in the title correlated, because they go hand in hand. We have not yet figured out the mechanistic basis for this. It's important to realize. So this is the part I want to skip, because it's an elaboration of a technical thing to study metabolism in mixed cell populations at the single cell level. If there's interest in that, we can go back to that after the talk, I would propose. Okay, so there's 2 slides for that. And because I want to mention this, because I think it's very exciting and new. I've told you already in the beginning that the glutamine is important for in the CLL lymph nodes. And when we were giving this talk at one time at the conference, there was a hematologist came to me, and she said, well, we should work together, because it's known in CLL, the standard glucose PET tracing does not work very well. And this is illustrated over here. If you do CLL imaging, in part A, we see an indolent CLL at diagnosis. There's nothing lights up. It seems, so this is the standard glucose PET tracing. If the cells transit to accelerated CLL in picture B, or in the Richter transformation, which is a highly aggressive form of CLL and resembles actually more DLBCL, you can see that now the glucose PET tracing can image those cells, whereas the majority of CLL cases are not visible by PET tracing. And this fits well with what I've just shown you, is that in the lymph nodes, the CLL cells actually use glutamine rather than glucose. So the idea was to use glutamine PET tracing. And this is not invented by us. It was already done for brain tumors, for example, which can also not be visualized by glucose tracing. So the chemistry for that was known, and we started collaborating with chemists and the hematologist José Saltra in order to figure out if we could make fluor-labeled glutamine and then use that to image CLL cells. And the data that you see over here are indeed, again, the labeling of CLL cells stimulated after CD40 stimulation, which we know increases their glutamine uptake. And this can be blocked with this inhibitor again, showing specificity. So we now had proof that we can use this fluor-labeled glutamine to label CLL cells, and this is in a culture. And recently we finally started using mice to do PET imaging. And this is a very famous mouse model called the TCL1 mouse, already published in 2002, and is adopted all over the world as a very good model for CLL in mice. And what we do in this kind of we do adoptive transfer. So we have TCL1 clones in the freezer, we inject them into mice, and then they have outgrowth of leukemic cells, as you can see over here, in a couple of weeks, instead of months to a year, which it takes in the original TCL1 model. So you see the spleen size in the TCL1 cells and the spleen weight. This is all sort of standard TCL1 leukemia. Oh, I think I'm going back. Yes. And here's actually the latest results, which we are very happy with. So now we started doing the glutamine and glucose tracing in these mice, and these graphs over here on the left are a bit confusing because the glucose is taken up in the spleen in normal mice and also in the TCL1 mice, but there the ratios are reverted, and this is plotted here in the middle, and the imaging is here on the right. So there's lots of absorption of the glutamine tracer by the kidney and also the bone. This is known. And on the left here is the control mouse, and here's the TCL1 mouse. And in this part over here is the spleen, which does not light up in the control mice, but does light up in the PET image of the TCL1 mouse, confirming that also in the mouse, the CLL cells use glutamine as an energy source, and much more so than glucose. So this brings me a little bit to the end, and I want to share some conclusions and perspectives with you. So I would like to stress that in the lymph node tumor microenvironment, we think the CLL cells are glutamine addicted. As I've shown, it has effects on mTOR and protein translation. For the T cells, what we've shown is that there are two types of dysfunction that seem to be linked with reduced mitochondrial activity. This last part I haven't been able to show you today, but so we'll skip this. And lastly, the perspectives we think we can perhaps target therapeutically glutamine import in CLL as a additional therapy for the current, actually quite good therapies that there are, but for example, in resistant venetoclax or ibrutinib resistance. Restoring T cell function of the CAR T cell therapy using, for example, TOR inhibitors or ACT inhibitors. And for diagnostics, we are in the process of developing glutamine PET tracers. And based on the results I've shown you, our institute is starting a small-scale clinical trial the end of this year to see the validity of the glutamine PET tracer. So I hope I've not confused you too much with all these data. I would like to thank you for your attention and just want to highlight the people and the names on the left and the pictures over here of the group, and also the collaborators, the original collaborators from Southampton for the CLL glutamine studies that I showed you in the beginning. We rely very heavily on a very efficient biobank team. The names are over here. We collaborate intensely with the metabolomics facility in our institute, and with the important collaboration also for the PET tracer project. The names are mentioned over here. And for the things I didn't address for the single cell metabolic analyses, we collaborate with chemical biology in Leiden. I thank you for your attention, and I'm open for questions.
Thank you, Eric. There is indeed one first question from the audience. Do you anticipate that the metabolic targeting will become a standard component of CLL treatment, or is it maybe it is a little bit far from now, but what's your opinion on that?
Right. Well, I think there is room at least to explore it. So what I try to tell you is that we think there is at least academic interest to study the validity of to use additional glutamine blocking to enhance the efficacy of current targeted therapies like venetoclax or ibrutinib. And also in the view of the resistance that develops in a fraction of patients. That is one thing. And like I said, the diagnostics is something that is perhaps very interesting to do. Because, for example, it is known that venetoclax is, of course, a very successful drug. But I think many clinicians will agree that there are usual residual lymph nodes in these patients, and of course the idea there is that is the source of resistant cells that eventually will emerge. And if we can image these things better, these lymph node sites better, then we might be able to offer more therapy, is the idea.
Thank you. Thank you very much. We have another question, which is much more perspective. What do you see as the most promising area of research in CLL metabolism over the next 5 years?
Oh, yeah, like I think I just mentioned this already. So there's part of our group is working on studying the T cell metabolic defects, also with the idea to assist the T cells in becoming more functional. And this goes also for CAR T cells, for example, that are, if you take T cells from CLL patients, the CAR T cells that you make from these are also not very good. And that is something part of our group is trying to enhance, for example, by interfering with protein kinase signaling, protein translation pathways, and so forth. That's something that's actively being pursued.
Thank you. Thank you. If I don't see other question, but I have both two questions, maybe mechanistic question. You look at the impact on chromatin of metabolic deregulation that you observed. Did you look at acetyl deposition in the chromatin of the CLL cells? And the other question is, I didn't get on your presentation if you have any clue about the cause of the mitochondria alteration that you see in CLL T cells. Is it the first step before allowing the CLL rise, or is it caused directly by the CLL cells?
Yeah. So the first part of your question about the chromatin, I didn't quite get. I didn't talk about chromatin. But
when I hear about metabolic reprogramming and the regulation, I think about chromatin. Do you see the impact on chromatin, on acetyl, on methylation?
Oh, okay, I get it. Yes, no, that is aspect we haven't studied. So it's the brief answer. I know that, of course, you're right. There is a connection there, but it's something we haven't studied. And the second part is when it is indeed the dysfunction that we observe depends on the presence of the CLL cells. If we for many of the studies that we do, for example, the seahorse, we have to separate them because it has to be pure cells in the seahorse. And so that's a lot of work to isolate pure CLL T cells and then put them in the seahorse. But you still see, although there are no CLL cells present, they are still sort of imprinted with this dysfunction. In the patients, if you treat them with ibrutinib, but it has been studied in our group more with venetoclax. If you give patients venetoclax, then the CLL cells go away, the immune system slowly regenerates or gets better as well. And if you look later in successful treatments, then these T cells are functional again. So it is not long lasting, if that is what you mean. Yeah, it depends on the presence of the CLL cells, but how this works is not really known. Also, your question of the mechanism of the T cell dysfunction. Well, what I've tried to make clear, but perhaps was not completely successful in, is that we so far seem to exclude options mostly. We tried import of glutamine in the cells. That's not it. It's also not the import of glutamine in the mitochondria. So I didn't show you all of it, but we striped out most sort of easy explanations. And for the moment, but we still haven't solved it yet. We are in this. We found that the shape of the mitochondria and also the ultrastructure under the EM, I haven't had time to show you these, is also altered in CLL T cells. So,
And sorry, excuse me, did you investigate the number of mitochondria per cell? Is it a question about the mass of mitochondria? I mean,
yeah, so that is altered as well. The mass seems to be increased, but they're sort of they're smaller and fatter. So to summarize, so I can go. Well, it's a lot of slides back, but yeah, so less branching. They're compact, and from the EM, from the top of my head, what I can say is these famous cristae that are in mitochondria. So these foldings of inside the mitochondria is also more disorganized in CLL T cells if you do EM pictures.
but indeed, I think that one of the most fascinating aspects of your current work is that we are almost at the point of understanding why CAR T cells have failed so much in CLL for 15 years. And because, I mean, among all B-cell lymphoma, T cells of CLL patients are by far the most, struck by the disease. They are completely not frozen, it's not the word, but they are really, really different. And we see that in patients, the type of infections, etc., etc. I think that maybe we are, with your work, starting to understand why we failed. And again, you straight out a lot of options, but we are still not understanding a lot of mitochondria, of the biology of mitochondria. And so, as you are an immunologist, my question would be, are you aware of other diseases, maybe not cancer, or chronic infection, viral diseases, I don't know, chronic CMV latency, where T cells resemble that, or it's really CLL specific?
It's a good question. What we studied in the past a bit, is that indeed the CMV T cells in CLL patients are increased in numbers, and it was always thought that it was due to improper control of the CMV, but that actually didn't yield an answer, and it is not the cause for the immune dysfunction, we think, because there are patients, of course, who are CMV negative, and they have the same dysfunction. I think in our institute, we also have a group that works on HIV, and there you see also a sort of T cell aging, is what they call it. And I think there might be a resemblance more with the T cell defect that you see on. It's not so much on people that develop HIV, but people treated with HIV therapeutics. So on what is it? They call it ART, I think.
Yeah.
Yes. So these
antiretroviral therapy.
Yes, and these people have what they call an aged immune system. But it's difficult to compare directly with CLL because these people are usually also very old. So what I showed you for healthy donor T cells, if I showed you data, they were age matched. So
Okay.
the difference is not due to younger versus old. Yeah.
Maybe I don't see any other question, but I have maybe a more translational question. Did you look at the preclinical data of V9302? Toxic? Is it feasible in your mice model?
It is, well, actually, we learned from this molecule from a study of a solid cancer model in mice. I believe it was a lung cancer. And it's in one of the slides as well. So that's when we first learned, there is a molecule that inhibits a glutamine import, because this whole field is a bit new for us. And based on this type of insight, more for solid cancers, there are indeed companies that are making prodrugs that resemble this inhibitor. Glutamine metabolism inhibitors have been under development for quite a while, we've learned by now. But most of these interfere with the conversion, for example, for glutamine to glutamate. They're called glutaminase inhibitors, and they I think, not so much under development, but the inhibitors of the import are of clinical interest. This is what I know, especially for solid cancers as well, because also these are glutamine addicted, not so much glycolytic.
Since the V9302 compound is far from specific, as you've shown, are you aware of maybe SLIC1A5 degraders like we have now, BTK degraders? Or it could be maybe much more helping us understand, at least in vitro, what's the different impact of a different, amino acid transporters.
No, I don't know, but the biology of these importers is very interesting, because we know also that it's one of the things that we saw in the beginning. If you stimulate the CLL cells, but also T cells, the importer goes up in expression, both on RNA and protein level. So these cells, B CLL cells, but also T cells, they increase the expression and import of not only glutamine but also other amino acids. It's just that in our experiments, the T cells don't seem to depend on it as much as the CLL cells do. So of course, both mice and men have more than just T cells, but at least the T cells do not seem to be affected by this inhibitor in terms of function. I've shown you proliferation, but we also measured function in presence of this inhibitor, and the T cell function does not seem to be affected in laboratory tests. We don't know the long term. But it's not something that we are not into drug development. We use these tools to study in the lab, but I think it's interesting for supportive therapies perhaps in the future.
And have you noticed any maybe differences in terms of glutamine addiction across different CLL specimen? I mean, for instance, NOTCH1 mutated, TP53
Ah,
mutated, according to the mutational status
so
of VH sequences also.
yeah.
Yeah.
Now it's a good point. I have to say that we applied for funding to do this kind of research, but this was not approved. So it's an interest, but we don't have the money to do it. I think you can appreciate the problem.
We do.
Yeah.
And have you any insight of a metabolic programming of B and T cells in Richter transformation, or absolutely not?
Yeah, that's a very interesting point. What I showed you actually, what the Richter cells do, they become much more aggressive, but they increase the glucose metabolism, as you could see on the PET scan, because Richter is visible in a glucose PET scan, and it also more resembles DLBCL. I mean, you are the clinician, but this is what I learned. But the metabolism, Richter is hardly untreatable, so it would be very interesting, I think, to study it. But of course, it is quite rare, and it's very difficult to obtain viable cells from Richter. If you have a source that you can provide us with viable Richter cells, we would love to study those. In our institute, we don't have them. They have microscopy slides from the pathology.
Yeah.
So actual activity you cannot measure anymore. So you have to do spatial transcriptomics, these kind of things. And I'm pretty sure that people will be doing that as we speak.
Yeah, I don't know any additional question.
Okay.
You have one.
Oh, getting late.
No. 7:08.
Well, I have a dinner date with my family, so I won't mind closing the session. Thank you
Sure.
for the opportunity. I would like to, but that's perhaps not for the audience, but I would like to get in touch about putting lots of unpublished work on YouTube. I just realized that this is perhaps we should take out some part of the presentation.
Of course.
Okay, I'll be in touch.
Sure.
Because it might be a bit of a risk these days.
And I think it's our turn to thank you very much for this beautiful talk.
Thank you.
I would just remind that you can subscribe and view later the Red Talk, and that you can go to see the other series. And we really thank you for your presentation. And if you have any comment, please, in the future, you can send us.
Okay. Thank you for the opportunity to share our work, and I wish you all a good night.
Have a nice family weekend, Eric.
Thank you. And enjoy Krakow.
I will.
Okay.
I will drink a beer with Anton.
Anton is there? Yeah? Okay. Yeah. Say hello to him, and I'm signing off now. Okay? Thanks a lot.
And
see you later sometime.
Yes.
How do I exit? Here. Bye-bye.
Bye. Hop.